And welcome back. Thank you so much, Cheryl, for such an informative talk. That was really, really interesting. I think Julie's going to lead off with her first question.
Yep. So one of the questions that came in was, you explained that stress alters learning and memory based on factors like arousal and timing. And to that list of factors, could you talk more about maybe how an individual's affective state or personality or even coping styles could affect?
Absolutely. So I didn't even talk about affective state, and that involves the amygdala. And the amygdala is absolutely key, and I'm going to talk about one study that wasn't mine. It was my colleague, Dave Diamond. His name comes up again. What I thought was a pretty brilliant study, and what he did was, for rats, a cat is a really robust stressor. You don't even have to have the cat touch it. It could just be like moseying around, and some people use odor, but so he uses a cat and he just pays it to hang around, and that causes a robust stress response, and that impairs these animals' memory. So he did it before and then had them learn a task and assess the memory, and they're impaired. He had them learn it and gave them the cat afterwards, and then assess memory, they're still impaired. And he gave the cat before the memory, and they're impaired. But what was unique about his study is he said, what else could raise stress steroid levels? Now we know that predator raises stress steroid levels. What else could? Sex. So what he did was he gave that rat access to a sexually receptive female, mated. Guess what? Cortisol levels elevated. And what did he find with memory? Not impaired. So certainly, affective state, emotional state plays a key role, but so too does the neural circuitry that's engaged. And what I'd like to argue is that with the predator, a certain set of neural circuitry was engaged. You had the stress steroid response, and that led to a detriment on their memory. Certainly, stress hormones were elevated with the rat mating with a receptive female, and it's important it's a receptive female because they'll fight otherwise. So in this case, you had the stress steroids elevated, but the neural circuitry is a very different one in that case. So it's a whole nother topic, but absolutely, I didn't want to play that down, but it's relevant, and I think it has a lot to do with the development of post-traumatic stress disorder, which, again, I didn't have time to talk about.
One of the things that we find in canine science is we're often trying to be informed, I guess, by research that happens in the human field, but we're very wary of being anthropomorphic as well. And so we don't want to over-infer. And I guess I was wondering your thoughts on how we can, I guess, be informed with the benefit of having the humans able to verbalize their experience of stress and how that relates to physiological correlates with the physiology that we see in dogs, but also, I guess, not throw the stress research baby out with the bathwater, if you will, by wanting to avoid being anthropomorphic.
Absolutely. So one of the reasons why the National Institute of Health was interested in funding my research was, first, National Institute of Health, what they do is they fund human health in the United States. That's what their interest is. And National Science Foundation funds more basic science and training. So why would the National Institute of Health be interested in funding my science? The chronic stress that we look at produces outcomes in animals that have a lot of parallels, in my case, with what we call depressive-like behavior. I wouldn't begin to say it's depression because depression is characterized by feelings, and we can't ask that in a rodent. But we can hone in on various symptoms, and cognition is one of them. And the nice thing about animal research is that we can begin to look at the various neural circuitry and symptoms, and perhaps by a portfolio of different kinds of research, we could get a better understanding of the whole characteristic in the human depression case. So with animals, when we look at a depressive-like behavior would be anhedonia, the lack of interest in something that they would find rewarding, is what we call it in rats. So sucrose, they love sugar. And so if we do any kind of manipulation where their interest in sugar goes down, that is what we call depressive-like. So that would be the reward pathway, for example. Also, they could show anxiety-like behavior where they will avoid certain potentially dangerous situations. And so in that case, we could look at how they behave in an open field. I kind of described that, where they'll stay along the perimeter. Another one is called the elevated plus maze, where they have 2 enclosed walls and then 2 open arms, and it's elevated. And rats, when they walk in those open arms, they could peer down and see that they're at a height, and that evokes a reaction in them, and they will avoid those open arms. And so you could look at how a treatment might alter that behavior, and that would be a measure of anxiety. So you could look at portfolios to begin to understand how various manipulations and treatments could help shift the physiology one way or the other.
Okay, thank you. I was just wondering if, there's this difference between acute and chronic stress, but is it sometimes hard to tease them apart, to say where does one end and where does one begin?
Absolutely. How do you tease apart the stress response? In fact, one of the things that I am finding very intriguing is I think, and I have a model where I use a chronic stress and overlay that with an acute stress as a model of PTSD. And so to answer your question, I'll take just a little bit of time. So with our chronic stress model, Miles already touched on this, where we put rodents in these kind of confined spaces, and they don't like it, so they'll have a stress response. And then we let them out and repeat this daily, and that creates the changes in the brain that I talked about. But that physiology that retracts these dendrites in the hippocampus, if I take that animal and then put it in a new environment where they have to learn a tone and foot shock, so brand new environment, brand new stressor, so that would be acute for most animals, but for this animal, they just had this chronic stress background. We put them in there. They actually acquire the task faster, so they learn the tone, foot shock cueing faster. They don't extinguish as readily, so they resist extinction compared to our controls who will extinguish more readily. And then if they do extinguish, they'll forget they've extinguished, and they'll be freezing and showing a robust stress response to the initial stimuli compared to the controls. So to me, that's a portfolio of post-traumatic stress disorder. So I think the acute and chronic is a great one, and we're all exposed to various stressors throughout the day. I think it's more how we react to those and our coping strategies that are in place, how we decompress. I like watching some programs on TV. I also like reading. I'm in a book club with Miles and a couple of our friends. So there are different ways that people could do that. And as a consequence, some of those stressors that I experience, I would like to think are not. They're just acute stressors. So it's a challenge, but I don't think I've quite answered it because it's kind of hard to do.
I love that your personal enrichment involves trash TV.
But also, so there are those physiological changes, but do you notice behavioral markers also to differ between acute and chronic stress?
Great question. So yes, absolutely, the behavioral markers. So with an acute stress, when we have a rodent who's presented with the first time a predator, a cat, it might try to evade it, and depending on the closeness of it, sometimes it makes sense for it to freeze. For animals that might have undergone a lot of chronic stress, they might go through what you call learned helplessness, where at some point they don't show the kind of shifting in behaviors. One structure I kind of tapped into was the striatum. It's a structure that's important for habit-based motor programs. So when we're learning to drive a car or ride a bike, you can recall, if you could remember that time as you're sitting and trying to drive the car, how you're focused, and someone starts talking to you, and you're like, stop, don't talk, I've got to concentrate. And you got to pick someone up at the airport, and you're like, oh, because you have to pay attention to all these signs, and you're just driving the car, and everything's so new. Now we're just like, la, 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 we don't even have to think about it. We could live in Manhattan for years and then immediately get into a car that we haven't been in for years and start driving. So the beginning of the driving is your prefrontal cortex. You got to have your attention, you've got to be attuned. And then after a while, it moves into a motor program, the striatum. The striatum actually is shifted in the part that's engaged under chronic stress. And it's thought that when individuals are presented with a stressor, it is in their best interest to do something that's tried and true. And so they default to the habit-based programs as opposed to something that's a little bit more flexible and uncertain.
That's excellent, thank you. Chronic stress and recovery via the enrichment, exercise and antidepressants have been investigated in terms of spatial memory performance, but how about its relationship with other cognitive states or types of cognitive states? Can we extrapolate to these other areas?
I actually haven't done, I've done, not with the antidepressants, but other people have looked at the prefrontal cortex, and a colleague of mine, Kara Wellman, is looking at that, and it's quite exciting. We started with the hippocampus because of some of the work that Miles touched on earlier with some of the changes that are happening there that was demonstrated in the 80s. But it wasn't until, I would say, late 90s, early 2000, where people began to look in the prefrontal cortex. And we don't have to extrapolate. Studies are being done in that area that we do see remodeling. In fact, one study shows dendritic retraction happening in as little as 7 days, and one showed that all you had to do is just inject the animal, not even give them corticosterone, this is rats, and that was sufficient to cause remodeling. So it's exquisitely sensitive. And that's important because when I mentioned the lizard brain, this is the emotions, the amygdala, and that is what guides us to react to these situations. It's our prefrontal cortex that says, hey, it's no big deal. That person who cut me off probably is racing to the hospital because he's trying to meet up with his wife. And when we think about that, we're like, oh, we want that person to make it safely. But if we use our prefrontal cortex to kind of justify and kind of calm ourselves down, it's easy. But when it's compromised, if it's our dendrites are remodeled so that they're not getting the effective inputs, then our amygdala is kind of taking over, and we're more reactive, more emotionally reactive than we probably would have been if we weren't under chronic stress.
Yeah. Fantastic.
We just have a little bit of time left, but looking forward, what research are you excited about?
We're doing two things, and what I haven't told you is that I have one graduate student who's interested in understanding the mechanisms of recovery. And we could block it. We could block it by stopping one protein from being expressed in the hippocampus. It's brain-derived neurotropic factor. We call it BDNF, and again, I look at it and I'm just so happy. But we could actually stop that protein from being expressed. We don't block it completely. We kind of just downregulate it, and we prevent recovery. And that's amazing. So we're interested in that story. And then the other story my other graduate student is interested in is this two-hit hypothesis of we have chronic stress, and then when we're presented with a very emotionally arousing experience of foot shock in our rodents, how can we attenuate those robust memories so they're just a memory but not an emotionally arousing one?
Okay, yeah, fantastic. Well, thank you so much, Cheryl, for your time. We look forward to you joining us on the panel later on this afternoon.
Thank you.
We'll be taking a short break, and we'll be returning as per the schedule in a little while. We'll see you all again soon.
Thanks.